So we were learning about aquatic respiratory organs. We mentioned the abdominal gill. Next, continuation. The caudal gills. Most of the Zygoptera nymphs bear the medial gill representing an appendix dorsalis and two lateral, the cerci. The caudal gills are filamentous and hairy dipetral structures. They are modified into swollen sarcoid gills or passing lamella gills and laminae. The gills are internally highly truncated and entire internal cavity is filled with tracheal system, nerves, blood channels and the alveolar tissue. The tracheal gill in the dragonfly nymphs, the anterior two-third part of the rectum is modified into a barrel-like chamber, and the rectal wall epithelium is differentiated into thick and thin regions alternating with each other. There are 6 thick epithelia, and each is differentiated into basal thick pads and distal gill filaments. There are the 6 longitudinal rectal pads. Each rectal pad is composed of alike 2 padlets, and each padlet laterally bears obliquely overlapping flattened gills. The gills are richly supplied with the tracheoles arising from efferent branches of the longitudinal tracheal trunk. The entering tracheae from opposite sides form a complex of loops within the gills. The tracheal chamber is quadrant anteriorly as well as posteriorly by well-developed valves, and it is innervated by a strong strands of longitudinal muscles and diffuse circular muscle fibers. The gill leaflets may be simple type of gills, undulating or papillate structures. They may be double type, duplex type gills, elongated filamentous, implicated type. Besides the nymphs of Anisoptera, the rectal gills are also known to occur in some Diptera larvae. The spiracular gills, spiracles of one or more pairs, bear long filament-like processes in the aquatic forms of beetle and that of Diptera families. Then blood gills, tubular or digitiform, irreversible structures occur at the anal extremity in 4 to 6 numbers in larvae of Trichoptera and 2 pairs on penultimate segments and 2 pairs on anal end in Chironomus Diptera larvae. They are more osmoregulatory than the respiratory structures. In physical gills, the hair absorb and store the air for inspiration, forming the physical gills. They are capable of absorbing oxygen from the aqueous media and are found in the hind legs of aquatic bugs. They can store air for a considerable long time. In some aquatic larvae and pupae of Diptera and Coleoptera, the spiracles are situated on sharply pointed processes which penetrate the air-containing cavities of submerged parts of the aquatic. Some aquatic insects breathe on oxygen taken from the atmospheric air. These use a special device which brings contact with some spiracles to the atmospheric air. They are found in respiratory tubes, metaphorus, modified hairy antennae. Crystalis contains respiratory cycles and quillicets their spiracles at the apex of cycles. In Notonecta, the hairs are concentrated around the spiracles, the hydrofuge hair. They are hydrophile on one side and hydrofuge on the other. As they touch part of the left wing of hair gets lower by suspension. The air stays with the communication with the hairs become hydrofuge on immersion but hydrophile on withdrawal from the latter due to reversal in orientation of polar molecule. In Notonecta, there is a fine pile of erect hygrofuge hair which holds between them a layer of air like velvet when immersed in water. Similar type of water film is also formed on ventral surface of body of Coryxa and Nepa. In aquatic, Coleoptera like Chemonia and Elmis are long hydrofuge hairs are distributed all over the surface of the body which are bent over the tips. They hold a thin film of air. Plastron, permanent water film in the beetles. and some other organisms. The air film is also held by the hydrophobic hairs that it cannot be replaced by water, and the insect become independent of the atmospheric air. It can live for months below the water surface if it is well aerated. This very thin, firmly held permanent layer of gas is termed plastron. In modeling, the gas is negligible, but interface is held in position by surface forces in such a way that its volume remains constant. Functionally, it resembles a tracheal gill more closely than an air sac. The plastron is well developed in the notochord bug. Most of the body surface covered with a very fine plaster held in position by an apicular hair. Spiracles are protected by a screen of long flat wings. These set with fine hairs, they open into a chamber from which radiating canals run forward in the form of four. Those channels communicate with the exterior with the minute opening along the course. The whole structure is being lined by the The next, physiology of gill and plastron respiration. Tracheal gills. Burrowing ephemeropteran larvae such as Ephemera and Hexagenia take oxygen through the abdominal gills, and about 45 to 47 of total oxygen uptake is carried out by them at VO2 of 6 cm. The frequency of gill beating is inversely proportional to the environmental VO2. These larvae behave as respiratory conformers in the absence of substrate and as respiratory regulators in the critical level at about VO2 of 1 cm in the presence of optimal substrate. In this ephemerid larvae, the lateral abdominal gills of all 7 pairs are composed of double lamina to provide greater surface area, which is they live in the lakes and ponds. The gills beat rhythmically at low O2 concentrations and intermittently in high concentrations. Chiron and Leptocellulidia larvae are in fact respiratory regulators at a critical level at above 1.5 and 2.5 PUCM. CO2 respectively at 100 cm1 of the environment. In beetles and Rhytrogena ephemerid larvae living in flowing water, the abdominal gills do not beat and larvae can only tolerate oxygen concentrations below 4 CUCM. The larvae are conformers. The gills play a little role in oxygen uptake as they are single lamellate and are less resistant to the low environment PO2. In Zygopteran larvae, 3 caudal gills are developed to become lamellate or sarcoid type and are either simplex or duplex in organization. They are the sole respiratory organs and function in low environmental PO2. They contain 32 to 60% of total oxygen uptake in Synagrion living in ponds and lakes. Rest of the oxygen uptake is carried by the winged insects. They are effectively respiratory conformers, capable of regulating their metabolism above oxygen concentration of 60% saturation. In low oxygen containing environment, the larvae spread their gills and sacs and move to the water. Shallow cells. The larvae low oxygen concentration and raise in temperature accelerated intervals, ventilatory resistance. Overall phase of increase proportionally with increase in oxygen concentration and decrease of the body weight. The carbon dioxide output increases in concentration and decreases with size. The larvae may gain to rocket and even be found here in larvae. Others obtain oxygen through at variable rate, and the Trichopteran larvae seem to exclusively be adapted for cutaneous respiration, in which the tracheal gills play a little role. Spirocular adaptations in aquatic insects. In some diptera larvae, peristigmatic glands discharge oily secretion and provide hygrofuge region around the spiracles, which allows air but prevents entry of water into the spiracles. In Notonectans, some diphtheria larvae, spiracles are encircled by the hydrofuge hairs which curve over the spiracles while the animal is submerged in water and prevent entry of water but spread widely as soon as the animal moves to water surface because of mostly the effect of surface tension. In some aquatic mosquito larvae, functional spiracles are situated at the tip of siphon and taking up oxygen from air, while in some aquatic larvae, the siphon penetrates the gas spaces of plant roots. The gas gills, physical, they are temporarily or permanent gas gills. The temporary gas gills carry an air bubble when the insect is submerged and extract oxygen from the surrounding water. Permanent gas gills or plastrons are also like that.